Chemotherapy is designed to destroy. It aims to wipe out rapidly dividing cancer cells with ruthless efficiency, and often it succeeds, at least at first. Tumors shrink. Scans look clean. The patient goes into remission. But then sometimes the cancer comes back. Stronger, stealthier, and more resistant than before. While genetic mutations and surviving cancer stem cells are part of the usual explanation, a new suspect has entered the scene: senescent cells. And not just any senescent cells, specific subtypes of them, lurking in the post-chemo microenvironment, potentially orchestrating the perfect storm for relapse. Senescence is often described as a protective mechanism, a cellular safety net that halts division in response to stress, damage, or aging. In theory, this is good. When chemotherapy blasts a tissue, many normal cells respond by entering senescence rather than mutating into new cancers. They stop proliferating and, ideally, get cleared by the immune system. But the reality is more complicated. These senescent survivors often stick around, secreting a toxic brew of inflammatory factors, growth signals, and proteases known as the SASP, the senescence-associated secretory phenotype. And in the context of cancer, the SASP can be a problem. Instead of quietly retiring, some senescent cells act more like angry neighbors with a megaphone and a suspiciously large compost pile. They stir up inflammation, degrade nearby tissue, and most critically, create a microenvironment that supports tumor cell regrowth. This becomes especially dangerous when residual cancer cells, cells that survived chemotherapy through dormancy or resistance, find themselves bathed in a cocktail of growth factors like IL-6, IL-8, VEGF, and amphoregulin. These molecules don't just attract immune cells or remodel the stroma, they can actually help awaken dormant tumor cells and encourage them to proliferate again. But not all senescent cells are doing this. Some may be relatively quiet, anti-proliferative, or even supportive of immune surveillance. Others might lean heavily into fibrosis or vascular remodeling. This is why the idea of senescent cell subtypes has become crucial. The post-chemo tissue landscape isn't just damaged, it's full of diverse senescent populations, each with different roles. Some are drivers of relapse, others might be innocent bystanders, or even temporary guardians. The challenge is to figure out who's who. Thanks to single-cell sequencing and machine learning tools, we can now begin to map out these senescent cell subtypes in cancer-adjacent tissues. In breast cancer, for example, senescent fibroblasts with high IL-6 and MMP expression have been shown to promote metastatic outgrowth. In prostate cancer, senescent epithelial cells left behind after treatment may alter hormone signaling and affect recurrence. In blood cancers, senescent immune cells may actually suppress T-cell function, dulling the body's ability to spot residual malignant cells. The picture that emerges is complex, but actionable. Different senescent cell types create different post-treatment risks, and understanding that heterogeneity is key to managing long-term outcomes. That's where predictive senescence profiling enters the picture. Imagine collecting tumor-adjacent tissue after chemotherapy and identifying which senescent cell subtypes are present. Is the SCSP profile skewed toward immunosuppression, angiogenesis, epithelial mesenchymal transition? Each of these could signal a different relapse risk and potentially guide follow-up therapy. Instead of waiting for recurrence, oncologists could act preemptively, applying targeted senolytics or SCSP inhibitors tailored to the specific threat landscape. Senolytics, in particular, offer a promising path forward. Early studies suggest that clearing chemotherapy-induced senescent cells can reduce metastasis and improve survival, at least in animal models. But again, specificity matters. A broad senolytic might kill helpful or neutral senescent cells along with the harmful ones. Ideally, we need senolytics that are subtype selective, targeting only the pro-tumorigenic senescent cells identified via surface markers, gene expression profiles, or even spatial location within the tissue. There's also the intriguing possibility of combining senotherapies with immune checkpoint inhibitors or vaccines. If we can modulate the senescent cell burden to reduce immune suppression, we might enhance the body's own ability to patrol for residual cancer. Some researchers are even exploring the use of senescence biomarkers as early relapse indicators, detecting shifts in the SASP signature in blood or tissue as a kind of relapse radar, months before traditional imaging could catch anything. So while chemotherapy may be the first wave of attack, what comes next is just as important. The senescent cells it leaves behind aren't just debris. They're active participants in the biological aftermath.
Some are peacekeepers.
Others are saboteurs. And if we can learn to read their signals, map their subtypes, and intervene precisely, we may finally be able to turn remission into something more durable, less uncertain, and ultimately more curable. In the war against cancer, it's not just about what we destroy. It's about what's left behind and how we handle the ghosts.